Hello, thanks a lot for the introduction, and thanks a lot also for the invitation. So I will, originally I said I would be talking about discovering disease effects in medical imaging data, and this will be part of the talk, but we had some really interesting results recently, and as I was making the talk, I realized that it might be more interesting for you to give you a general overview of what we are currently working on in our lab. And so the sort of unifying topic is that we really try to look at things in a probabilistic way and approach medical imaging problems from that point of view. So to give you a quick overview, first I will be talking about discovering disease effects using this Wasserstein gun approach that I will explain in a second. I will then show you a recent technique that we developed to do MR reconstruction using variational autoencoders. And lastly, I will talk a little bit about a weakly supervised learning approach, which is based on a flavor of expectation maximization. So just very briefly about myself. I started with my master's at ETH. I then briefly went to the surgical planning laboratory where I met Klaus Meyer-Hein, which is my connection to Heidelberg. I then did my PhD in London and a postdoc also in London. And now I came full circle and I'm at ETH again in the computer vision laboratory. I will skip this. Okay, so the first section of this talk is work on visual feature attribution using Wasserstein guns. It was joint work with those people here. So the goal is, let's say we're given a data set that has different classes of images. Let's say, for example, healthy images and images with a disease. But we know nothing else. We only know the class and the intensities. How can we figure out which pixels in the image belong to, let's say, a certain disease? And as I said, we have no pixel level information available in that data set. So I would say the sort of classical way, perhaps, to do this, how it is commonly approached, is to train a neural network classifier to predict a label. So this is some previous work of ours where we were trying to predict fetal standard planes. And once you have the prediction, you can kind of go back and there are certain algorithms that then allow you to highlight the pixels in the input image which affected this prediction the most. So in this grayscale image over there, here you can kind of see those pixels were important for classifying this as a fetal 4-chamber cardiac view, for instance. Okay. And it wasn't just us. Many people choose this general approach. However, we found some problems with this. And to motivate this to you, why this approach may fail sometime, consider this toy classification problem. So we have data with two classes. Let me see if I can make this work here. Okay, it doesn't, whoops, really work. Anyways, so we have a label 0, which you can consider as healthy images, and they are it's just a noisy background. And then we have label 1, which you can consider as diseased images, and they have two subtypes. So they always have a square in the middle, but sometimes they have a square in the lower right corner, and sometimes they have a square in the upper left corner. But the central square is always there. Okay, so now if you train a classifier to predict label 1 or label 0, but not the subtype, and you look at this saliency algorithm that I explained before, and see which pixels influence the classification, you can kind of see down here the saliency map. As training goes on, the box in the upper corner becomes less and less important. So the classifier is only focusing on the middle box, And what's more, it's only focusing on one edge of the middle box. And from a classifier interpretation point of view, this makes a lot of sense, because in order to distinguish the two classes, all you need to know is one edge of the central box. However, from a sort of finding the pixels that belong to this class point of view, this is not what we want, right? Because essentially we would only find this one edge rather than the two boxes. Okay. So this is why we decided to approach this whole problem from another perspective, not involving classifiers at all, or not in this classical way. So to quickly introduce some formalism. So let's say we have two images. We have a distribution of healthy images, C0, and we have a distribution of sick images, C1. So in this case, they are brain MRI images that are healthy and brain MRI images that have Alzheimer's disease. So we pose the problem as in the following way. We say, can we find a deterministic function, M, that if we add it to an image of the diseased class, will make the image look like an image from the healthy class. And this map will in some way contain the difference that distinguishes this image from a healthy image. So it will contain the disease effects. So to formalize this a little bit more, so let's say we sample an image from the diseased class, C1. And if we input this into the x plus into this equation, I think maybe I want to walk around a little bit after all. So we input this here to get a y. Then this y will be distributed according to some new probability distribution, q. And what we want is this q to be as similar to the healthy distribution as possible. So we want some distance function that allows us to measure the distance between the image of real healthy images and the distribution of generated healthy images, Q. And of course, you might have guessed, generative adversarial networks provide a framework to do this. Specifically in this work, we used Wasserstein GANs, which are a flavor of GANs, if you don't know it, which can be shown to minimize the Wasserstein distance between two distributions. So the framework works like this to show it graphically one more time. So we sample an image from the sick class. We add something to it to make it appear like this. And then we say, this image here should be indistinguishable from a normal, real, healthy image. And the equation below is the equation for the Wasserstein GAN. That's just putting this figure into equations. And that's not the only thing we want. In addition to this GAN loss, we also want to preserve the identity of the subject we're transforming. So we put this L1 regularization loss on the mask generator function, saying the changes that we make to the image should be as small as possible. So the total optimization problem is we look for the parameters theta of our mask generator function that minimize the GAN loss and the regularization loss above. So if we look at this toy example with the boxes again, the 3 rows are 3 different examples. You can see that this class of gradient-based method that I was already showing in the beginning produces these very spurious saliency maps. There is another approach called class activation mappings, where you construct your neural network in a specific way to get these kinds of maps. It's a little bit better, but you can see that the resolution of the predictions is very, very low. And then lastly, our proposed method manages to find these boxes very nicely. And what's also nice to note is that once we have trained this mask generator function, we can actually generate this image in milliseconds, right? It's just a forward pass through our mask generator network. Okay, so of course we weren't interested only on applying this to boxes. We wanted to apply it to something hopefully clinically meaningful. Specifically, we focused on neuroimaging data, and even more specifically, we focused on this problem of Alzheimer's disease. So Alzheimer's disease, you may be familiar with, is a neurodegenerative disease that happens to many people later, in the later stages of life, which causes these atrophies in different brain regions. Especially this region of the hippocampus shrinks a lot, which is the region which is required for memory and for navigation skills and things like this. But also you can see other parts like here and around the ventricles shrink a lot. So can we identify these changes in a population of images using the method I described? So for those of you who work on medical imaging data, a few details about the experiment setup. So we worked on this ADNI data set, which is very big. It consists of, well, we used around 6,000 images of around 1,300 subjects. And a part of them had mild cognitive impairment, which is like a precursor to Alzheimer's disease. And a third of the images had, those patients had Alzheimer's disease. And so as I said, the objective is to generate a subject-specific disease map for a person with Alzheimer's, highlighting which regions of this specific patient have been affected by Alzheimer's disease. Another thing that I briefly want to mention is that we had a small number of subjects that had MCI, and we acquired an image was acquired at the stage of MCI, and then later the patient went on to get Alzheimer's disease, and we also had an image at that stage. So we could use those paired images to obtain a sort of ground truth in quotation marks about the disease effect. However, we didn't use this pairing for training, and it's not necessary for training. This was just for evaluation. So these are the results. So again, the 3 rows are 3 different examples, and the columns are the different methods we investigated. You can again see that these gradient-based techniques produce very spurious predictions. Class activation mappings produces very poorly resolved estimations. The last column is the ground truth in quotation marks. And you can see that our proposed method matches these ground truths very nicely. Another thing in this plot that's interesting to note is that the subjects are sorted by increasing severeness of the disease. And you can see that up here, very few regions get identified, whereas down here, where the patient is at a much later stage in Alzheimer's, the algorithm correctly identifies larger areas of the brain as diseased. So it doesn't just always toss on the same effects. It can really figure out what specifically this subject, what effects this subject has. It's occasionally underestimating things a bit, like in this case, but overall it performed quite nicely. You can look at the close-up of the hippocampus, which is this region that's involved in memory formation. So this is a real MCI image, and this is the same patient after they got Alzheimer's disease, and you can see the shrinkage of the hippocampus and the kind of shrinkage of the tissue around the ventricles. And this is then the sort of fake MCI image which we generated. And you can see that it sort of manages to reverse the atrophy around the ventricles. I should say the contours are the same in all 3 plots. Those are just for reference. And also, if you look around here, it manages to sort of grow the hippocampus a little bit. And also the generated images look very realistic.
Okay. So a few words on what is really new in our work with respect to a lot of the previous work on this topic. So the classic approach to sort of discovering disease effects in neuroimaging populations is voxel-based morphometry. It's been around for quite a long time. So the general idea is very simple. You align all your images to a template, and then you check for each pixel the distribution of your two populations. And if the distributions are significantly different, then you say this voxel was important for this disease. So a little, interestingly, one of the first studies this method was applied to was to study the brains of London taxi cab drivers. And it turns out that when you compare the brains of taxi cab drivers to normal people, well, not that taxi cab drivers are not normal people, but people who don't drive taxis, you can see that the taxi drivers have significantly larger hippocampal areas. And the hypothesis, of course, is because they have to remember so many street names and be able to navigate the city from memory. However, the drawback of this is that you only get a map like this for the whole population, not for a specific taxi driver, but for the entire population of taxi drivers. Whereas we were interested in generating subject-specific maps, which could really, you could imagine in a clinical setting, tell a doctor specifically which area of the brain has been affected by a disease. Furthermore, subject-specific maps could also be used to sort of stratify disease effects. So it is suspected that Alzheimer's disease is actually a collection of multiple disease. So you could be looking for, different subtypes in that way. Another drawback of voxel-based morphometry is that it requires registration. And of course, if you align your images perfectly, there will be no more differences between the two populations you're analyzing. So you need to register them enough that the subject differences are gone, but the disease effects are still there. And of course, that's very heuristic, like how much registration is enough. There's no good answer to this. And of course, registration also fails and takes a really long time. And yeah, so the advantage of our method is that it doesn't rely on registration. And the advantage of voxel-based morphometry is that you can make it work with relatively small data sets of like 50 to 100 images, while our method really requires very big data sets. But on the other hand, voxel-based morphometry is also very hard to make work with very big data sets if you have them, because you need to register all your images. Okay. So to conclude this bit, as I was saying, we showed that our method can produce these subject-specific disease effect maps, which we think could be interesting for discovering disease biomarkers and for disentangling subtypes of different diseases. And we also had some preliminary results where we applied it to other data, for example, chest X-ray data, where it can also be interpreted as a sort of weakly supervised localization method, where you can find, masses and things like this in chest X-ray images. And if you are interested, the code for this work is available on GitHub. Okay. All right. The second piece of work I wanted to talk about is in collaboration with these guys, especially Kerem, who is the first author. And the topic of this was MRI image reconstruction using a variational autoencoder. So to motivate this quickly to you, as probably many of you know, if you acquire an MRI image, you really acquire the data in what is called the K-space, in a sort of frequency domain, and then you perform a Fourier transform to get to the image. Okay. However, often it can take a very long time to sample every frequency in the K-space. So people are interested in undersampling the K-space to reduce the number of parts you have to acquire, speed up the acquisition. However, if you do this naively, you of course get these aliasing artifacts that get more severe the more you undersample your case space. So what we are interested in is acquiring an undersampled case space, but then still be able to reconstruct the original image without these aliasing artifacts. And we decided to approach this problem from a Bayesian point of view. Let me see if actually that's maybe a good solution. Okay. So in this equation here,
Y is the case space data, M is the image, and E is the sort of function that generates the case space from our image. E consists of an undersampling operator, a Fourier operator, and a coil sensitivity mapping operator. So we know how to get from the image to the case space. We don't know how to go the other way, though. Also, we say here, in addition to that, we have some measurement noise on top of this. And this measurement noise could be caused, by thermal noise in the scanner and things like this. So we can write this like this. So the case space data, given the image data, is distributed around this mean here, and the noise of this variable here, so the observation noise. Okay. So really what we are interested in is the opposite, right? We want the image given the case space. So we can go back to the Bayesian theorem and rewrite this conditional probability like this. We can further say, if we're just interested in the map estimate, we don't need to worry about the normalizing P of Y. And lastly, we can say, if we can also optimize the log of the whole equation, which yields these two terms. And perhaps, if you are familiar with this sort of reconstruction problem, this is the way how you usually see it. You have a data term and you have a regularization term. Okay, so let's get a little bit of intuition for this. So we know we have a likelihood function that tells us which images are likely with this case space data. So let's say they live in this ball here. Yeah? The other thing we have is a prior model, which gives us the sort of manifold of all possible MRI images, P of M. And really what we want is the intersection of the two. So we want an image that's consistent with our case space data, that's also on the manifold of realistic MRI images. And lots of approaches exist to model this P of M. One of the simplest ones is you just say the reconstructed image should have a small number of high frequency components. This is called a total variation regularizer. Or you can try to learn this distribution using kernel density estimation, or using something like PCA. Or more recently, people try to learn the distribution of patches of these images using dictionary learning. So here are some results using those techniques. If we zoom in a little bit, you can see that so this is the original image, this is the undersampled image with the artifacts. Total variation produces these very cartoon-like images. And dictionary learning, for instance, produces actually, much nicer results than total variation. Manages to retrieve those lobes quite nicely and so on. Okay, so our contribution here is to learn this distribution of MRI images using a variational autoencoder. For those of you who don't know about variational autoencoders, it's sort of assuming that your high-dimensional data in reality is caused by a much smaller number of latent variables, Z, and you learn sort of the mapping from your latent variable Z to your high-dimensional images. And of course, if you do the whole thing, this will end up in an autoencoder structure. And one more comment. It turned out that modeling the whole MRI image was too high-dimensional to work for now, so we also resorted to modeling just patches rather than the whole image. So here you can see some real patches on the left, and some patches generated using a variational autoencoder, and some patches generated using PCA. You can see that the variational autoencoder manages to capture these lobe structures and things like this. It's also a little bit overly smooth and cartoon-like, but at least the general structure it's capturing is right. Okay. So now, let's say we learned our P of M. So let's assume we now know this guy, right? So that's been trained, that's done. So now we want to reconstruct an image. So let's go back to the optimization problem. So how do we do this? So we want to optimize this equation here. However, the prior is only approximate because, as we will see later, you don't actually learn P of M. You can only learn like a lower bound. So this doesn't actually have an exact intersection. However, you can differentiate this prior. So we can actually look in which direction in image space we should walk to get closer to the manifold of real images. So how we do this is in a two-step optimization. So we start with some reconstruction consistent with the k-space data that would have lots of aliasing artifacts and not be close to the manifold of MRI images. We calculate this derivative, and we walk a little bit towards the manifold. Okay, now we have a realistic image that's not so consistent with our k-space data anymore. So we project it back using this equation onto satisfying k-space, onto images that are consistent with the k-space. And then we continue doing this a couple of times. So, and yeah, the last point, when you calculate the VAE, you don't actually get the real density P of M. You get a lower bound, which is usually referred to as the evidence lower bound, the elbow. But we say that the derivative of the elbow approximates the derivative of the real density quite well. Okay. So this is what happens in these optimization steps. So you can see gradually it's kind of removing these aliasing artifacts. Let me restart it one more time. So these are the separate back and forth projections you're seeing now. Okay. So let's compare it again to the related works. So you can see that, so this is our proposed method. It manages to capture these lobes here almost even a bit more nicely than this dictionary learning techniques. And this is a different work that's also based on neural networks, and this also outperforms this work a little bit. You can also quantify this in terms of root mean squared error and contrast to noise ratio for two different undersampling factors. And we see that in both cases, the proposed method produces better results than the methods we compare to. Okay, now, so far, I haven't really talked about other works who use neural networks for MRI reconstruction. And so what most other people do who use neural networks is they simply learn the mapping from k-space to image space as a supervised learning problem. And there is, this I think works pretty well, but there is a big problem with this. And the problem is once you learn this network, you're fixed to a certain acquisition setup. So when you used an undersampling rate of 2, this network won't be able to do reconstruction with an undersampling rate of 3, for instance. Or if you used a certain undersampling pattern, it won't work with a different undersampling pattern. And our proposed method doesn't have this problem because you learn the density of MRI patches in the image space. So you can use whatever undersampling pattern you want and just use this as a prior for realistic images. So to illustrate this, we did this experiment where we did reconstructions with different undersampling rates. And so this is what you get, without correction, Cartesian and radial. And this is what you get after reconstruction with our method. And exactly the same method. You don't need to retrain anything. So you can see in both cases the reconstructions are quite nice. Okay. You can also do it with complex data. And one last thing that we thought was really cool is, so we trained the whole thing on healthy images from the HCP dataset, if you're familiar with this. But then we decided to also test it with some images with Alzheimer's disease from the ADNI dataset, which we heard about in the last part of the talk. And you can see they are very different images. They have, for one, still have the skull. They have different contrast properties. And also they have these white matter lesions here, for example, and here. So these are lesions that occur to people who have Alzheimer's disease. And what was really interesting to observe is that even though the method was not trained on this data, it still managed to reconstruct the images to a satisfying degree, including these lesions here and here. Those surprising results that we were very happy about. Okay. All right, so on to the last section of my talk. Yes?
Or maybe a quick question to the last part.
Yeah, sure.
Have you ever tried applying your VA GAN on this problem?
My VA GAN? This, you mean how?
Like the conditioned on this image with the aliasing errors, and then the output would be a clean one?
Yeah, we did try that, in fact. So what happens is also what you, I guess, observe in many natural image problems with GANs. So when you look at the generated brains at first glance, they look really good, really sharp, and you're like, wow. But then when you look closer, you see it's making up brain structures. So it's sometimes adding new brain lobes that aren't supposed to be there, and things like that. So, I mean, I'm sure there would be some way to combine it, but like the naive GAN approach doesn't immediately produce good results on this. And of course the cool thing about the VAE is that you can actually sort of model this P of M. So you can kind of really understand what's going on, because in the optimization later, you're just moving towards realistic images. Okay. All right, so the last part of my talk is about a method that we presented recently at the workshop at MICAI. And it's about training a neural network with scribble supervision alone. So the motivation for this problem is that, of course, as you all know, training neural networks requires loads of data. But especially when working with medical data, it's very expensive to annotate those data, because only medically trained professionals can do it. And especially for pixel-wise segmentations, it takes those doctors a very long time that they need for annotation that they could probably be spending for something better. So our sort of research question here was, can we find a way of training neural networks based on a weaker type of annotation, in this case scribbles, that is much quicker to acquire? And so by scribbles, I mean just drawing these lines across the structures of interest. Rather, this would be, for comparison, a pixel-wise annotation. So what we want is to train a segmentation network with only annotations like this, such that after training, when we test a new image, we can obtain a full segmentation mask without the requirement for any additional scribbles or anything. So all the network ever sees during training is scribbles, and during testing, we wanted to produce full segmentation masks. Okay. How do we do this? First, some related work. So, yeah, we are not the first ones to think about this. For example, Reichel et al. Did something similar using bounding boxes. However, we found bounding boxes in this particular case a little bit awkward because the structures overlap a bit, and it's not immediately clear what you should do with these large areas here that are part of all of the structures. And there is also a little bit of work on scribble supervision on natural images. This work is based on superpixels, and superpixels just are known to not work so well on medical images in our experience. So to our knowledge, this is the first work to try this problem with scribbles on medical data. Okay, so to formalize again the problem a little bit, let's say X are our pixel values, intensity values. Z hat are the scribbled pixels. You can remember them because it has a scribble on top. And Z are the labels of the unannotated pixels. And then theta are our network parameters. So what we want is a maximum likelihood solution of this distribution here. And if Z hat was not unknown, we could simply do the standard thing, and it would be a simple neural network training. So if you model this by a Bernoulli distribution and take the log of it, you end up with the cross entropy as a loss function. However, we cannot do this because we are missing the values Z here. So what this is a maximum likelihood problem with missing data. So it's kind of natural to think about expectation maximization. So the intuition of expectation maximization, if you are not familiar with it, is to start with an initial guess for your parameters phi. You then use that approximation, that guess to approximate a distribution of Z. And then using that distribution of Z, you find a better value of theta. And then you iterate like this. And it's shown that this will at least converge to a local maximum solution. So this is, again, putting it into a picture. So we train a network, we estimate this distribution of our labels, we relabel our training set, and we retrain the network, and we do this in a loop a couple of times. Okay, so to formulate this properly in EM, we added a little log here. So we want to maximize this probability distribution with respect to theta. And as is standard in expectation maximization, because we don't have Z, we instead take the expected value of this probability distribution here with respect to our best guess of Z, which is given by this equation using the old parameters. Then once we formulate this equation, we can optimize for a new theta that will increase this function. So to make life a bit easier for us, we used what is called a hard EM approximation, where we say, actually, we don't want to model this whole distribution here. We're just going to assume it's very peaked. And we're just going to model it as a single delta function, basically. And to find the location of this delta function, we need to find the in the E step, this Z that maximizes this probability distribution. And we can rewrite it as the joint probability distribution because PZ hat doesn't depend on Z. So, and why do we want to do this? We want to do this because for the joint probability distribution, we can basically use a very efficient optimization algorithm out of the box. And this is a conditional random field, a CRF. So you can rewrite this problem here basically as this optimization problem. And to make lives easier for us, we sort of started treating the labeled and unlabeled pixels in a similar way. So if you don't know a CRF, what it is, basically you first make a fully connected graph of all your pixels. And then you say each pixel wants to be something. The unlabeled pixels here want to be something that depends on the network output. And the labeled pixels, they just want to be what the annotators scribbled them to be. Yeah, so they are unchangeable, basically. But then also everything wants to be similar to its neighbor. So you have these pairwise terms here between scribbled and scribbled and unscribbled pixels and all the combinations. And this pairwise term, we simply chose the same one as described in this work here. So I won't go into details. And what's cool is this can be solved very efficiently, again, using the technique described in this paper. And you can get a new labeling Z star. Okay, once we have this labeling Z star, we want to plug it into our Q function. So we use this factorization for our joint probability. And this is the Q function from before, but we plug in the mode of this distribution we found. So using the sifting property, this equation up here, you get these two terms. And we notice that this term doesn't depend on theta. So when we try to optimize this term, all we end up is this. So basically this equation here. And you may recognize this again as a normal neural network training. So this is just finding the parameters that maximize the labels we found in the relabeling step. Okay. So that was a lot of math. So let's quickly just go back to the picture and get an intuition for this. So in the E step, we take some parameters that we found earlier, and we use the network to relabel our training set. Then in the M step, we use this relabeled training data set to train a new neural network based on these relabeled data. And we keep doing this. So there is something I haven't mentioned yet. This is that you may have seen that these are actually not our scribbles, right, here. Our scribbles are here, but as an initial guess, what we do is we use this random walk segmentation algorithm, which was proposed by Leo Grady, to flesh out these scribbles. And the random walk segmentation algorithm, similar to neural networks, gives you a sort of per pixel probability of a label. And we thresholded it at a very, very high stage, such that ideally all the pixels that are actually a label should be definitely that label. And everything that's black in this figure, we treat as uncertain. And uncertain in this context means at these points, we multiply the cross-entropy loss in the neural network training with 0. So in those locations, the network can do whatever it wants. It doesn't get penalized. And this is only in the first iteration. Okay. All right, and then we had, in addition to this base algorithm, we had a little of small extensions that we tried. One thing was to, instead of using this dense CRF, as described earlier, we used this CRF RNN network that was proposed recently, which does the same thing, but you can add it onto the network as an additional module. And the cool thing about this is that it automatically learns the CRF hyperparameters. You don't need to find them in a cross-validation or something beforehand. Okay. Then the second little extension was to use this technique called MC dropout. Was proposed by Yarin Gal to make sure that our relabeling is actually certain, that we don't propagate errors. So how we did this is we introduced dropout into our segmentation network during training and during relabeling. Then during the relabeling step, we do a couple of forward passes with dropout on. And so this means for each pixel and each label, we get some distribution of activations. We look at the distribution of the most likely pixel label and at the distribution of the second most likely label. And if these two distributions are not significantly different as measured by a t-test, then we reset that label to uncertain, meaning the network in these locations can again do whatever it wants in the subsequent iteration. Okay, so we experimented on two data sets. One was a prostate data set, and one was a cardiac segmentation data set. Notice that also there's always also a scribble for the background, so we just drew it around the structure of interest. Okay, so here are the quantitative results. First of all, if you just train a network one single time on these, random walked scribbles, you get a sort of lower baseline, and we can see that including this what we call recursion, this retraining and retraining, we get a little boost in the average DICE score of these two data sets. So these are DICE scores. These are 3 cardiac structures, and these are 2 different prostate structures. Okay. Then we can see by adding CRF, we get another little boost on this data set, not on this data set, interestingly. And then adding these little extensions seems to help also in some cases. And admittedly, the improvements are, not always significant, but in the end, we found that these two combinations, one with the CRF RNN module and the separate CRF and the uncertainty estimation, produced on average the best results. And here we also see as an upper baseline what happens when you train a network on the full segmentation. And we can see that it's actually not too far away in terms of DICE scores. So we're getting pretty close. Here are some qualitative results. On the cardiac data set, you can see that it captures this shape of the heart very, very well. The only mistakes are here little mistakes at the corners of things, at the edges of the structures, which is also what we would expect. Then the prostate data set is much more challenging. Here, the differences are a bit larger between the ground truth and these two methods. However, still, you can see that it definitely captures the right shape of the structures. So, in conclusion, what we really found remarkable is that we had a remarkably small degradation of performance with respect to the full supervision. So it was only around 3% worse on cardiac data and only around 4.5% worse on prostate data than a fully supervised network. So we believe that for some studies, this may be an acceptable error margin. And of course, some interesting future directions may be mixed supervision, for instance. What happens if you include a very small number of fully annotated cases and then annotate the large numbers using only scribbles? And we have a feeling that this might, close this gap even more. Okay. And yeah, that's all I have. Thanks for your attention. Yes?
And what happens if you go 2 slides back where you compare the fully trained RNN?
You mean this or the figures?
This.
Yeah.
So fully supervised means that you have the same, basically the same algorithm, but you know the labels of each pixel, or you use a completely different algorithm?
It's the same network architecture and so on, but since you have all the labels, there is no unknown labels. You can just train it like a neural, it's just a normal neural network training.
Okay.
Yeah. Yes?
Like in the study, is there a takeaway, would you say, with respect to whether noisy labels hurt the training? Like so the base model plus uncertainty basically means you zero out uncertain parts of the segmentation, right? Like you explained.
Yeah.
So you reduce label noise during training. Is that correct?
Well, no, actually, so we treat the labels as the scribbled labels. We treat them as absolutely correct. So under no circumstances are they ever changed during training. So this uncertainty only applies to the newly relabeled things. And I see your point, and I think it's what you're probably trying to get at is that sometimes you may have scribbles that actually go across a boundary or something. And this is something we want to look into in future work. So right now, the scribbles were annotated in a way that didn't allow to go across boundaries. So we cheated a little bit in the generations of the scribbles. So they are like very, very good. And they follow exactly the full segmentation.
And do you have a feeling for how hurtful it is? Because generally deep learning seems to be very robust, remarkably robust to noise like that.
Yeah, I can only speculate, but this is also what I found in other projects, that you can really increase the label noise a lot and still get reasonable predictions. But we never try to quantify this so far. Yes?
Were the scribbles generated by people, or you had them from the full encode?
Yeah, it was generated by a student that we had. So how he wrote a little graphical user interface where he could draw scribbles on top of the images, but whenever a scribble went outside of the full segmentation mask, it got chopped off, basically. So, yes?
How much time do you save annotating scribbles instead of full mask? Because I assume in 3D, how do you do it in 3D then?
Yeah, I mean, so this was performed on so the data isn't 3D actually, but we performed it in a slice by slice fashion. So all the networks were in 2D. And so I mean, doctors obviously would annotate also in a slice by slice fashion, although they can look also at orthogonal planes if they want. But the idea of scribbles would be that they just go through the slices and draw a scribble on each one of them. And the student did a little study where he just tested himself how much time he saves doing the scribble with respect to doing a full annotation. And I cannot remember the number right now, but it's really a significant time saving. I think it's something like 7 times faster or something like this. I would need to look up the exact number, but it's really a big time saving. Yes?
I heard you tried a mixed approach where a student or a person would scribble, then the network would run, and then you would see the result of the network, and then you can scribble the rows that are not correct.
I mean like an interactive segmentation approach.
Iterative scribbling.
Yeah. So actually we have some other work from our group that looks at exactly this problem. However, this was in a completely different framework, so it's not necessarily related to this. So in this kind of framework, we have never looked at this, no. Oh yeah.
You said that you were only looking at one slice at a time.
Yeah.
And from manual labeling, I know that it's a lot easier to, for example, go back and forth between the slice you're currently working with and the one before or afterwards. did you also think about including more slices in like a single analysis in your network to, like, for example, look at the slice before and after?
Yeah, to sort of incorporate the 3D information into the training. No, it's an interesting point. We haven't really looked at it so far.
Also to like maybe reduce errors when there's one slice that's labeled slightly incorrectly.
Yeah.
And maybe.
Yeah. So I think it's like generally in neural networks, particularly on cardiac segmentation, I think it's been showed that if you have the full segmentation masks, doing it in 3D usually leads to improvements. On this particular data set, there are big stack misalignments. So it's actually hurtful to do it in 3D, but that's just a problem of the data set, not of, and yeah, so with respect to the scribbles, we would have to see whether the information is dense enough to take advantage in the, through plane direction or not. Yeah, it's an interesting point, but we haven't really gone into that direction yet. But thanks for the comment. Yeah.
Your first project, where
Yeah.
you generatively learn the difference mass between diseased and healthy brains.
Mm-hmm.
I was wondering how diverse the samples of your GAN are that you get, because I think typically people have found that the conditional GANs often collapse to a deterministic mapping, basically.
Mm-hmm.
And if they're diverse in this case, do you average over the samples to produce the maps that you showed?
So one thing to note here is that our mask generator function, this quickly, come here, where's the mask? This guy here is actually a deterministic function in our case. So so it's only a function of the input image. It doesn't have any like noise additionally to it or anything. So in that sense, it will always produce exactly the same image. That's sort of the extreme case. But practically, it doesn't matter so much because, so it would be nice to get this additional information of, of the spread of possible disease effects. Because, as you are saying, it's probably ambiguous in some ways. You don't know. But again, so far we have only tried it with this deterministic function, and we haven't added noise or anything at this point.
That's interesting. So in this particular case, couldn't you then have learned the very same thing with, say, a mean squared error loss or something? Because I think you showed examples of the ground truth. So assuming that for each image you also have the ground truth disease mask, could you have just learned it with a regular loss rather than an adversarial loss if you treat it deterministically?
Yeah, but we don't have paired data actually.
Ah, right.
So these paired images, so we used a small number of paired images to generate this, but we only had around, I think, 40 of them or something like this. Whereas the whole data set is thousands. So the cool thing about this is that you don't actually need corresponding data to learn it.
Makes sense.
Cool. Yes?
We use this to detect artifacts in the image pipeline. Because that's also not paired, like a motion artifact.
Yeah. You mean in general, or?
In general.
Yeah. So some collaborators of ours in London have applied this method to shadow detection in ultrasound. So they had some images with shadow and some images without shadow, and then basically used the same approach. And I think it produced okay results. And I think they published it later on, where they used this as a single step in a larger framework. But so there can be one problem with this, and this is the problem of like confounding factors. So in their case, some views had more shadows than other views, and so when you try to remove the shadows using the gun, you not only remove the shadows, but you also started changing it into another view, because one of the views just had more or less shadows than the other one. If that makes sense. So in some cases, to make it really robust, you would need to somehow take this confounding variable into account. Yes?
If I remember correctly, you used L1 regularizer as a constraint.
Yeah.
Have you also tried using like a cycle gun as a more loose constraint?
No, we haven't tried that yet. So this is like, personally, I find that going from a diseased image to a normal image is sort of the easier way to go, right? Because you can have many variations in the disease, but the healthy images are a bit more stable. So I'm kind of trying to avoid to go the other way. I don't want to be making up diseases because I feel like this is an ill-posed mapping somehow. But yeah, it's been frequently suggested actually, and it's also on the checklist to try this. So I don't know, these cycle guns seem to be able to do pretty cool things. So we will definitely look into this at some point. But my feeling is that I don't want to go the other way for this particular problem. Oh, yeah, sorry.
On your project with the MRI images?
Yeah. Also, you mean the reconstruction?
Exactly.
Yeah.
You were talking a lot about reconstruction in the image space, but it should be also possible to do the reconstruction in K-space. That means if you have done your sampled image in K-space, could you reconstruct the full image in K-space and then do the Fourier transform of that?
Let's see. Okay, I see what you mean. However, I mean, I think this could definitely work. We haven't tried it, but I think this could work. But the advantage of doing it in the image space is exactly that you get independent of the K-space. And this is advantageous for many reasons, because you get independent of the K-space pattern. But actually, if you learn from the full K-space, you would also, the undersampling pattern wouldn't matter, right? So that's a good question, actually. Yeah, we haven't tried that. Very good comment. Yes?
Was that on magnitude images, or was it complex?
So most of this that I showed was on magnitude images, but then the reviewers asked us whether it also works on complex images. So I went over this very quickly, but we extended it a little bit, where you, in addition to this, you also have a sort of a phase projection step in the optimization. And it also works. I think it's a little bit less robust to undersampling, but it still works. Yeah,
okay, great. Let's thank Christian for this very inspiring, very cool talk. Let's give him a round of applause. So before we go into the mingling phase where we'll have snacks and drinks, let me quickly show you what's in store for you guys in the next 1.5 months, I think, here at heidelberg.ai. Let me quickly flip up the slides for this. Just a second. Let's see. Cool, this works. All right, so the first one is a conference on AI in art and design. It'll be held on November 8th and 10th in Frankfurt at the Museum Angewandte Kunst. heidelberg.ai is partnering with a bunch of people that organize this. The first 2 days will be actually tutorials where you can, get your hands dirty and program something yourself. And November 10th we'll invite 6 different people from this space, AI in art and design. So we'll have, for example, one of the very prominent figures, Mario Klingemann, who I think was an artist in residence at Google, and we'll also have AI curators. So that should be a pretty interesting event. Next up, just shortly after, on November 12th, we will have Professor Michael Feind talk about how he uses AI in his company towards building an autonomous supply chain. Michael Feind was actually the supervisor of Paul and my master thesis, and from experience I can tell you you're in for a treat here. He's a very good speaker, very knowledgeable. Yeah, he has built up Blue Yonder, I think, in about 2008, and at that time deep learning was sort of in its infancy. It didn't really boom yet, but already then he built his company upon neural networks. And they've been recently acquired, so they're way past their startup phase. I think it'll be really interesting to listen to Michael. And then lastly, on November 26th, we'll have Marta Garnello, who is both a PhD student in London, but also a research scientist at Google DeepMind. And she will talk about 3 papers of hers, very recent ones, that all evolve around the concept of using generative models for few-shot prediction tasks. So again, I guess a very interesting talk, and yeah, we'd like to invite you for all 3 of these. All right, now lastly, we'd like to thank our research group, the Medical Image Computing Group here at the German Cancer Research Center, DKFZ, led by Klaus Meyer-Hein. We tucked on an image, so yeah, we're actually quite nice. We're looking for master's and PhD students. Yeah, apply if you're interested. We're doing cool stuff. Yeah, thanks for your time. And now we'd like to invite you for snacks. They're outside, wraps actually, and we also have, beverages and beer. Thanks for your time.